Well, good afternoon if you're based in Europe, and good morning if you're calling in from the United States. We're going to cover specifications for small molecule drug products today, which is a very large topic to cover in just 30 minutes. Obviously, we're not going to be covering it in depth, but I'm hoping to give you one or two important pointers along the way. What we will be discussing is current regulatory guidance, tests that are appropriate to different dosage forms, and we'll take a look at a couple of different dosage forms as well as looking at guidance that's generic to all drug dosage forms. Dissolution specifications for different solid oral drugs. Dissolution specifications also in the context of the biopharmaceutics classification system, which I'm sure some of you are already familiar with. There are a few things that have a significant influence on the safety and efficacy of drug products, and those include polymorphism, enantiomeric purity, drug-related impurities, and microbial contamination, so we'll spend some time looking at those. Specifications for combination drug products, particularly in the context of impurities, and how we go about setting specifications for impurities that derive from one or other of the particular drug substances present in combination products. And then specifications in the context of drug product stability. We don't have time to cover drug substance specifications, although of course those do have an important bearing on drug product specifications, the concepts of periodic or skip lot testing, in-process tests, design and development considerations, which are very much part now of quality by design, parametric release, which is using a parameter that we've monitored during the process to validate the fact that we've produced a batch of product that meets a particular quality attribute, and an example there might, for example, be recording the temperature of terminal sterilization in an autoclave as a means of demonstrating compliance with a sterility parameter, and then the impact of the drug substance on drug product specifications. All these are well covered in the guidance from the ICHQ 6A, which deals with specifications for drug products. The following ICH guidances are important references for specification setting. ICHQ 6A sets guidance on drug substance and drug product specifications. Q6B, by the way, deals with specifications for biological drugs, which we won't be discussing today. Q3B is ordinary impurities. Q3C is residual solvents. Q3D is elemental impurities. And there's a relatively recent guidance on genotoxic impurities, ICH-M7, that sets what's known as a threshold of toxicological concern for genotoxic impurities. And it's just worthwhile mentioning that residual solvents and elemental impurities are usually controlled in drug products by measuring them in the components that go into drug products. We'll look now at some regional guidances, the first from the United States FDA. And the first two guidances that are mentioned deal with scale-up and post-approval changes. That's what SUPAC stands for. So these are changes that we make to the drug product during the scale-up or after we've successfully registered our drug product with the FDA. And immediate release drug products, which is what the first guidance relates to, are drug products that are designed to release the active ingredient within 45 minutes. Modified release drug products include both extended release drug products, which are designed to release the drug over a longer period of time, or delayed release drug products, and those are normally formulated to survive passage through the stomach intact, commonly because the drug substance is unstable under acidic conditions. There are then a couple of guidances dealing with dissolution testing. The second of those is interesting. That's a relatively recent guidance, and it begins to introduce into regulatory thinking the sort of risk-based approach that was recommended in the ICH guidance, ICHQ 9, quality risk management. And it talks about class 1 and class 3 drugs as being of low risk. We'll come on to BCS class 1 and class 3 drugs a little bit later, but those two classes of drugs are considered a relatively low risk for dissolution. And I thought it was also worthwhile putting down a reference from the AAPS journal, a relatively recent reference dealing with dissolution testing for generic drugs from the FDA's perspective. We'll look now at European and World Health Organization guidances. So from the European Union, specifications and control tests on the finished product. It's quite an old guidance, but this is the document where we get in the European Union the requirement to control assay within 5% of label claim at release. Then there's guideline on the quality of oral modified release products, so that will be delayed release and extended release drug products. And the most recent one from this year is the reflection paper on dissolution specifications for generic oral immediate release products. 2 WHO documents are listed here. One deals with guidance on fixed dose combination products. So these are drug products that contain more than one active ingredient in a fixed proportion across the range of doses in which they're manufactured. And the second one is the WHO Expert Committee on Specifications for Pharmaceutical Preparations, which is general guidance. On now to tests for different dosage forms, and we'll start by looking at universal tests, and these are tests that are relevant to all dosage forms of drugs. We're not covering these comprehensively, we don't have time to do that. So if you need information on a particular dosage form that's not mentioned in this presentation, please look at ICHQ 6A. So the universal tests are description, which would include things like colour, shape and size. For example, we often produce tablets and capsules with different markings, different colours to denote different strengths. That's an important safety consideration so that we don't get mixed up between the strengths that we're taking. We are obliged to identify the drug substance in the drug product, and we can no longer do that for new applications on the basis just of a chromatographic retention time. We used to be able to do that, and if you look at some pharmacopoeial monographs, you'll see that there's a single identification test for the drug product which is based on chromatography, but now we need often a spectroscopic test to back that up. But we can perform the two tests simultaneously if we use a chromatographic method with a spectroscopic detector, for example a diode array detector, if that UV spectrum is sufficiently characteristic. The assay is a universal test. We've already mentioned the requirement in the European Union to meet plus or minus 5% of label claim at release. In the US that limit is commonly plus or minus 10%. And we need, of course, limits for impurities. And when we're talking about drug product impurities, we're not including in that category anything that's a drug substance synthetic impurity that isn't formed by drug substance degradation. So any process impurities, process intermediates that are carried over into the final drug substance that are not degradation products don't need to be controlled by the drug product impurity method. Tests for solid oral dosage forms such as tablets and capsules include dissolution and disintegration. There are some circumstances where we can use disintegration as a surrogate for dissolution. We'll talk about that a little bit later. The two physical parameters, hardness and friability, are commonly measured during manufacture and performed as in-process tests, so we don't often perform those as separate tests in the QC laboratory. Uniformity of dosage units is designed to make sure that we're producing a drug product that has a tight distribution of drug content around the label claim, and we can do this by measuring either the mass of the tablets or the mass of the capsule contents, or by performing an assay on individual dosage units to make sure that we are controlling that parameter tightly enough. And there are different requirements for both of those. We can use the drug content approach in every case. We can only use the mass approach if there's a large amount of drug substance in the drug product, and the size of the drug product is relatively large. We're normally interested in measuring the water content of our drug product. It affects chemical and physical stability commonly. And microbial purity is important, so we set limit tests for different types of microorganisms, including the absence of specified microorganisms. Parental dosage forms are those that we inject, and it's important that those are sterile and that we control the amount of endotoxins and pyrogens that would have an unwanted physiological effect on the patient. We try to exclude particulate matter from these dosage forms because of the potential of solid particles to block capillaries in the body, blood capillaries, and osmolarity is an important quality attribute where we have stated the tonicity of the drug product on the label. The way in which we set dissolution specifications varies depending on the way in which the drug product is designed. For immediate release drug products, we usually set a single time point specification, except where the drug dissolves very slowly, where we may want to set 2 time points, one early in the dissolution profile and one close to quantitative release, to make sure that we're characterizing that drug product dissolution profile adequately. For single point specifications, we normally take the sample between 30 minutes and 45 minutes, and we set a Q usually between 75% and 80% dissolved. If dissolution rate affects bioavailability, or things that we can do during the manufacturing process might affect the dissolution rate, it's important that our dissolution test is discriminating, by which we mean it's able to tell the difference between drugs with acceptable in vivo performance and those that don't have acceptable performance. So in this slide we're looking at an example of how we might use a parameter such as tablet hardness to set a specification for dissolution. Tablet hardness can change on stability storage, and it's also often a function of the compression force that we've used during manufacture of tablets. And the slowest dissolving drug product batch in this slide has unacceptable bioavailability. So we're going to want to make sure that we control the rate of dissolution so that it's more consistent with the other batches. So if we look at the amount dissolved after 30 minutes, if we set a dissolution specification of not less than 80% Q dissolved within 30 minutes, then we are adequately controlling the dissolution profile of the drug product batches that we're manufacturing. We can use disintegration as a substitute for dissolution if we can meet 3 criteria. Those criteria are that the drug is highly soluble over the whole of the physiological pH range, that dissolution is rapid, and that disintegration correlates with dissolution. And if we can answer yes to those 3 questions, we can use disintegration instead of dissolution. If not, we need to set a single point dissolution specification. Single time point specification. However, recent guidance from the FDA on the dissolution testing of BCS class 1 and class 3 drugs suggests that you can justify disintegration in place of dissolution, provided that those classes of drugs dissolve very rapidly, and the suggested disintegration specification is within 5 minutes in order to justify using disintegration in place of dissolution. So let's now consider dissolution specifications for modified release drug products. The first of those is delayed release, and we set 2 time points. Remember that delayed release drug products are designed to survive intact in the stomach. So the first specification time point is not more than 10% after 2 hours in acidic dissolution media, and there's then a buffer stage test where we want to check that we've got quantitative release under neutral pH, usually at pH 6.8, but some coating systems are designed to release at higher pH, so the pH that we test at will depend on whereabouts in the gastrointestinal tract the drug is designed to release. For extended release drug products, we need at least 3 time points. The first of those time points, typically between 1 and 2 hours, is an important safety control to make sure that we don't see dose dumping, which is where the drug releases all of its contents in the body very shortly after ingestion, and where that happens we can exceed a toxicological threshold. We can start to see some toxic side effects if we see dose dumping. And then the final 2 time points are designed to characterize the release profile of the drug product to make sure that it's consistent with what we've shown to be acceptable batches of drug product previously. We've mentioned the biopharmaceutics classification a couple of times before. Let's now define what we mean by the biopharmaceutics classification system. So it's a way of categorizing drugs in the context of their solubility and permeability. So solubility means solubility across the whole of the physiological pH range, that's pH 1.2 to 6.8. Permeability means the ability of the drug product to cross the intestinal membrane. And drugs in classes 1 and 3 are highly soluble. Drugs in classes 2 and 4 have poor solubility. Classes 3 and 4 have low permeability, so their rate of absorption in the body is generally slower than those with high permeability. And that's important because where we see dissolution as the rate limiting step to drug uptake, then obviously the dissolution test, the in vitro dissolution test, might correlate in some way with the rate of uptake of the drug in the body. But where permeability is the rate limiting step, we may well not see any such correlation. So to build on what we've just said, BCS class 1 drugs often dissolve so rapidly that it's difficult to characterize their dissolution profiles. Often the drug is released quantitatively within about 5 or 10 minutes. And in these cases, the in vivo absorption rate, the absorption rate in the body, is commonly controlled by the rate of gastric emptying. So the tablet dissolves rapidly in the stomach, but it doesn't become available for absorption until it enters the small intestine. And the rate at which it does that is controlled by the stomach emptying its contents then into the intestine. For BCS class 2 drugs, dissolution is the rate limiting step for in vivo absorption. So we might expect the dissolution profile to correlate with drug plasma concentration. And you may have heard the expression in vitro in vivo correlation, which is where we establish a quantitative correlation between in vitro dissolution and in vivo absorption, uptake of the drug in the body. And an in vitro in vivo correlation is commonly achieved for BCS class 2 drugs. Drugs in BCS classifications 3 and 4 have poor in vivo absorption kinetics because they're poorly permeable. And in these cases we can't usually predict what happens in the body by looking at the in vitro dissolution profile. We're going to consider now how we control polymorphism in drug products. Polymorphs are different ways of arranging the same molecule differently in a crystal lattice. So if you take a look at the diagram on the right hand side, the top two diagrams, polymorph 1 and polymorph 2, show different arrangements of the same molecule in a crystal lattice, giving rise to two polymorphs. In addition, we can form solvates if we have a solvent molecule included in a regular fashion in the crystal lattice. Where that solvent molecule is water, we call it, of course, a hydrate. If it's any other molecule, it's a solvate. And the control of polymorphism is important because different crystalline forms, and of course amorphous forms as well, exhibit different solubilities and therefore different dissolution rates, which can have an impact on the efficacy of the drug if one particular polymorph is much slower to dissolve than the other. We need to control polymorphism in drug products if, first of all, the API is capable of forming polymorphs, and the polymorphs have different properties, and the safety or efficacy of the drug product is affected by those different polymorphs. And where that's the case, then either the dissolution test must control the risk to the drug product, or the polymorphic form must be monitored during drug product stability to make sure that it doesn't change. Enantiomeric purity often has important efficacy and safety implications. Back in the 1950s there was a famous scandal involving the drug thalidomide. Only one of the enantiomeric forms of thalidomide had the pharmacological activity that was desirable. The other enantiomeric form resulted in birth defects from children born from women who'd taken thalidomide for morning sickness. So an important safety and efficacy concern. And this is how we go about controlling enantiomeric purity in drug products. The first question we need to ask is the drug substance chiral? If it isn't, we don't need to worry any further. If it is, but it's racemic, we need to consider verifying the chiral identity of that drug substance in the drug substance itself. If the drug substance is chiral but is present as a single enantiomeric form, then we need to test the drug substance for chiral identity, assay, and purity, and we need a similar test for the drug product. So now on to the control of ordinary impurities in drug products, and ordinary impurities are those that do not have any genotoxic potential. We'll cover genotoxic impurities later. So the first question we ask is, does degradation occur during drug product manufacture? If yes, we need to estimate the maximum increase in the impurity during manufacture, and then we go on to estimate the maximum increase in the degradation product at the end of the drug product's shelf life. We then set the maximum likely level as the drug substance acceptance criterion. That's the maximum likely level of the impurity at the end of the drug product shelf life, provided that the maximum likely level is greater than the qualified level. And the qualified level is the amount of impurity that we've shown to be safe through toxicological studies. And if the maximum likely level exceeds the qualified level, we need either to set the acceptance criterion to the qualified level, which means commonly we need to reduce the shelf life of the drug. We need to establish a new qualified level on the basis of fresh toxicological studies, or we need to change the storage conditions of our drug product so that the impurity doesn't grow as rapidly. And if the maximum likely level is less than the qualified level, we can simply set the acceptance criterion as the maximum likely level. Genotoxic impurities are controlled in a different way, so ICHM7 allows higher limits for short duration treatments. So if you look at that table, the limit that's on the right hand side, the 10-year or lifetime of the patient limit of 1.5 micrograms per day, is a toxicity based limit for whole lifetime exposures. That makes the assumption that the patient will be taking the drug throughout their lifetime. But you can see that where the duration of dosing is shorter, for example during a clinical study, we can justify a higher level for that genotoxic impurity. Which leads on to an interesting point. So if we can make this argument for genotoxic impurities on the basis of toxicological data, can we not do the same thing for ordinary impurities, where we're taking medicines for acute conditions over a much shorter period than the whole life dosing of the patient? Microbial contamination is controlled differently depending on whether the drug product has any antimicrobial activity. So that antimicrobial activity could arise from our use of a preservative in the formulation to inhibit microbial growth, or it may come about as a result of an inherent property of the drug product that inhibits microbial growth. So for a drug that contains an antimicrobial agent or has inherent antimicrobial activity, we establish limits for the preservative. We show what limits the preservative needs to be at to have antimicrobial effect. We establish the microbial limit acceptance criteria according to the United States Pharmacopoeia and its USP General Chapters 61 and 62 that are relevant here. And we perform microbial limit tests on each lot. If we can show that every lot of drug product that we produce consistently meets those acceptance criteria, we can potentially justify skip lot testing. That's testing on not every lot of drug product that we manufacture, or we could justify no testing at all. So the second case is where the drug has no antimicrobial agent added and has no inherent antimicrobial activity. So the first question to ask is our drug product dry? If it's a tablet or a capsule, a solid oral dosage form with purely dry ingredients, there may not be enough water in the drug product to support microbial growth. So if we can answer yes to that question, and we can show that there's evidence of the inhibition of microbial growth, in other words that the microbes don't grow in our drug product, we may be able to justify not performing microbial testing on that particular drug product. On the other hand, if it's a liquid dosage form, an aqueous dosage form, or there is evidence for microbial growth, we need, of course, to establish microbial acceptance criteria and show that we consistently meet those. Just going to the first point in this flow chart is the drug product a dry dosage form. The USP General chapter on water activity provides useful evidence to demonstrate the likely potential for microbial growth based on water activity. So water activity is the amount of water that is available to support microbial growth, as opposed to water that's locked up in, for example, water of crystallization and is not available for microbial growth. And this USP General chapter is a useful reference. It contains a table showing the minimum levels of water required to support the growth of particular microorganisms. So we can use that very often to support our strategy for microbial growth for dry drug products. So impurities in fixed dose combination products pose a particular challenge. Fixed dose combination products contain 2 or more active ingredients, and we've mentioned already the World Health Organization guidelines on fixed dose combination products. And the impurity acceptance criteria are expressed with reference to the parent API. In other words, we ideally need to be able to show which API the impurity has come from, not to the total content of APIs, and we then express that impurity as a percentage of the amount of parent API that's present. And if an impurity either results from the reaction between 2 or more APIs, or we can't show which API it's come from, its acceptance limits need to be expressed in terms of the worst case situation. That means if we know that a particular impurity comes from a reaction between 2 APIs, we express that impurity as a percentage of the lowest concentration API. If we're not sure where the impurity has come from, we need to express it as a percentage in relation to the API that's present in the lowest amount. Finally, we're going to talk about specifications and stability. The release specifications that we set for our drug product are there to safeguard the stability of the product to the end of its shelf life. So they're designed to protect the quality of the drug product all the way through to the end of shelf life. And when we're designing those specifications, we identify what's known as the shelf life limiting attribute, and that's the specification limit that is exceeded first on stability evaluation. So it's the specification attribute that causes our drug product to fail stability first. And often this is the principal degradation product, the degradation product that forms at the highest rate, or has perhaps the highest toxicological concern. And we can design packaging usually to protect the drug product from chemical or physical deterioration. We can protect from moisture using desiccant packs or particular moisture barriers. Different types of packaging have different moisture barrier properties. And also, if our drug product is sensitive to light, if we use an opaque package, we can offer usually adequate protection from light to safeguard the shelf life of that drug product. And we use the results from drug product stability studies under long-term conditions, so that's the conditions under which we're expecting the drug product to be stored, accelerated conditions for oral drug products that will commonly be 40 degrees C, 75% RH, and any stress conditions that we've used, for example, to look at degradation pathways for the drug. And we can use these various studies to establish a correlation between these parameters, such as moisture content and the degradation rate of the drug, and we will then base our specification for moisture content at drug product release on what we know about the degradation rate of the drug in the presence of moisture. So that brings us pretty much to the end of the available time. So unfortunately there isn't time for questions, but you can see my email address on this slide, and if you have any questions, please email them to me and I shall do my best to answer them. Thank you very much for your attention.